Hello everyone. In this video, we will try to understand the transition from yeast to Hyphal growth in Candida albicans. First, what is Candida albicans? Candida albicans is a type of fungal pathogen that leads to severe systemic infections that can be life-threatening and superficial mucosal conditions like vaginitis. Candida albicans also has the ability to transition from its yeast form to a Hyphal form. This transition is triggered by environmental cues and pathway signals that regulate the Hyphal morphogenesis. The characteristics of yeast and Hyphal are shown in the table below. So, in order for the Candida albicans to transit from yeast to Hyphal, or known as Hyphal morphogenesis process, it can be triggered by several environmental cues. Some examples include environment in the host niches, such as elevated temperature up to 37 degrees Celsius, presence of serum, elevated carbon dioxide levels, low glucose content, and pH condition, which is pH 7. Not only that, the presence of N-acetylglucosamine, amino acids sensing and quorum sensing molecules can also induce the Hyphal morphogenesis of Candida albicans. These conditions play an important role in the signaling pathway of the Hyphal initiation, which lead to the Hyphal morphogenesis. There are two pathways that hold importance for the transitional form from yeast to Hyphal, which are cyclic adenosine monophosphate protein kinase A pathway, CAMP-PKA, and mitogen activated protein kinase pathway, CEK-MAPK. The CAMP-PKA pathway plays a critical role in morphology, mitochondrial function, the development, glycogen synthesis, and energy metabolism of Candida albicans. All of those are possible by the production of UME6 protein, which is known to exhibit all the crucial functions mentioned before in the process of Hyphal elongation. Next is the CEK-MAPK pathway. The activation is triggered by various factors, including the surrounding extracellular matrix milieu, cellular injury to the cell wall, and the limited availability of nitrogen resources. When triggered, the pathway produces ACE2, a transcription factor that regulates transcription and performs a variety of tasks in cell division, Hyphal morphogenesis, and biofilm formation. Hyphal morphogenesis can be divided into two parts to make it easier, which are the initiation and elongation. During the initiation process, hyphae development begins with the septin ring formation, which acts as a scaffold. When a yeast cell meets all the requirements for Hyphal induction, the septin subunit, CDC3, CDC12, and SEP7, will form a stable core of the septin ring. Another subunit, CDC10, will become dynamic and form a shuttle between the septin ring and the cytoplasm for the preparation for nuclear division. The nucleus from the mother cell will migrate towards the septin ring within the developing hyphae, where the first nuclear division occur. This results in the production of two daughter nucleus, where one of the daughter cell will migrate back to the mother cells while the other will migrate to the apical compartment. The absence of any constriction or cell separation in Hyphal cells is due to the lack of the localization of CDC14, which is responsible to dephosphorylate MOB2-CBPKL1 complex, which activates a transcription gene for cell separation. Then, the septin ring will split into two rings and be maintained. Thus, the primary septum is formed, dividing the Hyphal compartments. The elongation process focuses on the polarization of the actin cytoskeleton. It plays an important role in Hyphal morphogenesis, as it maintains the directional growth of the hyphae by directing the vesicular flow for the tip expansion. The actin cytoskeleton is made up of two main components, which are actin cable and actin patches. Hyphal polarized growth are driven by the Spitzenkorper located at the tip of which are specific for hyphae compared to yeast and pseudohyphae cell, which were controlled by the polarisome complex. Both components are controlled by the same protein, SPA2, BNI1, and BUD6. As the septin ring forms and Hyphal growth continues, versicles that originate from the Golgi containing essential protein for the Hyphal growth will travel along the actin cable via the factor 5 myosin, MYO2, towards the Spitzenkorper where it will accumulate. Then, the accumulated secretory vesicle will be loaded on the actin cable, nucleated by the polarisome complex, specifically the BMI1, to the plasma membrane where they are docked with the exocyst. One of the exocyst subunits, SEC3, ensures that the secretion of vesicles happens predominantly at the tip to ensure optimum membrane material supplied for the development of new cell walls. This process is called exocytosis and will be counterbalanced by the endocytosis process, in which the sites are formed by the actin patches that occur through the clathrin-mediated endocytosis. Endocytosis maintains the polarity through the endocytic recycling of the polarity protein from the exocytosis process. The GT-PASI RSR1 present at the tip of expansion will be activated by the BUD2, which will localize the CDC24 at the tip in addition to the calcium bind on it, which aids in the polarity establishment. This process will continue as long as adequate supply is provided, and the requirement for hyphal elongation is fulfilled. In conclusion, the pathogenicity of Candida albicans depends on its ability to alternate between yeast and hyphal growth forms. Candida albicans also heavily relies on the mechanism of internal signal transduction, signaling pathways and external environmental signals that mimic the host niches to undergo the transition from yeast to hyphal form. That's all for now. Thank you.